Hello and welcome back to another Biology of Cells and Tissues video. Before we get started, leave a like on the video, subscribe if you haven't already, and again, always as we're going through, if you have comments, questions, concerns, anything like that, put them in the comments and I will answer them as quickly as I can. Okay, so talking about blood, getting through this material, the next couple of lessons, compared to the professor who kind of originally generally put this information together, I definitely did some rearranging as well as kind of adding some additional material as well that I know for a fact is going to continue to come up in the future. So we're going to see kind of how long this takes us. So we're going to talk about the blood in general, the major function characteristics, the major components. It's the origin of its tissue as well as the other connective tissues. Checking the composition function of plasma, also comparing that to serum. We're going to talk about the red blood cells and hemoglobin quite a bit. And then finally the white blood cells as well. So, as with all of our other connective tissues, blood is also coming from mesenchyme. If we stop and we think back to our embryology, which of our original 3 layers does the mesenchyme come from? It comes from the mesoderm. So all of our connective tissues are coming from the mesoderm. And with our blood, it comes from that mesenchyme. And we start off with a hematopoietic stem cell. Remember, heme, anytime you see the word heme or hema, anything like that, hemato, all of those kind of prefix, suffixes, all that, everything that like that means blood. Heme correlates to hemoglobin, which is blood, okay? From that stem cell, we get our different blood cells as well as red blood cells, white blood cells, and a whole bunch of different other things, platelets, monocytes, all that kind of good stuff. I didn't put in here red blood cells, erythrocytes, and there are a couple of other more specifics as well, but we don't really get into all of those, okay? So these are all the ones that we're going to be talking about in this PowerPoint. Platelets, also called thrombocytes, leukocytes, which are white blood cells, monocytes, which turn into macrophages, and then red blood cells, which are called erythrocytes. So functions of blood, some of these are a little bit obvious. Hopefully they're kind of a little bit, more common sense. Others we want to talk a little bit about. So the main function of blood is to deliver oxygen and the nutrients to the cells. All of our cells need oxygen to survive because this oxygen is used to produce ATP, which is the energy that that is required to essentially to do anything and everything in our body, which you'll talk about more about that in biochemistry. So it also transports the metabolic waste to the lungs and to the kidneys for elimination. Carbon dioxide, which is part of the energy process, goes to the lungs, and then all sorts of different kinds of waste actually goes to the kidney, which you'll get a lot more detailed into the kidney, kind of what goes down with that and how all of that happens, like what kind of waste products go through and how everything's filtered in trimester 3 in physiology 2. We also transport hormones from the endocrine organs to the specific target organs and other parts of the bodies in order to maintain homeostasis. Again, that's more Tri 3, a little bit this trimester in biochem in this class, more so a little bit in Tri 2, and then more in Tri 3, Tri 4, and throughout. You'll be talking about hormones a lot because a lot of our pathology has to do with hormones. Blood also regulates our body temperature, maintains osmotic pressure being the pressure of the fluid, so like how much fluid is in our vessels versus outside, dealing with like swelling, that kind of stuff, making sure that there is the correct amount of fluid in all the different parts of the body. And also its job is to prevent hemorrhage, so massive blood loss, by forming clots. That's those platelets, those thrombocytes. I'm going to say that word a lot, thrombocytes, because it's often a word that students, for some reason, does not stick in their head. So we want to make sure that we really, really nail down this terminology, so that way, we can, when we go to test, we're not relying on, we're not messing up on the terminology. We're not messing up on the terminology. Like, we know the information, but the vocabulary is getting us stuck or getting us the questions wrong. Okay, and then finally, immune system, the defense, are those white blood cells. So lots and lots of function. So the good thing to note and understand that this whole system is closed. Obviously, we don't have blood leaking out or coming in from anywhere else. It's all closed. The nutrients come in through the GI tract. The nutrients are delivered to the cells. Waste is removed through the lungs, through the kidneys, and through the lungs, and it just continues to go around and around and around. Oxygen comes in through the lungs, and we continue this whole cycle. Okay, it's a circulatory closed cycle, or closed system. We'll talk a little about this a little bit more later. We have organic nutrients. There's carbohydrates, amino acids, proteins, which we've talked about already. There's fats, cholesterol, vitamins, all that kind of good stuff. We also have non-protein nitrogenous substances. A lot of this, again, kind of coming and stemming from amino acids, from our proteins: urea, uric acid, creatine, and ammonium salts. This slide isn't super important as far as test material goes. Don't worry too much about this specifically. Know that it's a closed system, and where the nutrients come in and where the waste goes out. Here's the hormone pathway coming in into the bloodstream. Again, down below as well, I have lots of extra definitions, kind of some other stuff to start to understand this stuff. If you'll be good with what is specifically on the slides. However, if you want a little bit more detailed description of what's going on, or start, learning more of the terminology for future, that's down there at the bottom for you to look at. Here's maintaining the body temperature. This is done by absorbing heat in certain areas and then distributing it as well. It's specifically controlled through vasodilation, vasoconstriction. So parts of the body dilate or constrict. Parts of the vessels and parts of the body either dilate or constrict to control how much heat is lost. For example, if we're working out and we're really, really warm, the vessels near the top of our skin tend to dilate more. That's why when you see people exercise, they can get red, more of a red or a pink color, because they have more blood at the surface of their skin in order to let off some of that heat. As well, it can dilate. That's why you think your fingers, your toes, your extremities, those tend to get cold first, or get the most cold, because your body wants to keep what's most important alive. So your heart, your lungs, your brain. So it's going to constrict the vessels and all of the outside parts of the body in order to keep the core nice and warm. Now, clot formation, again, those platelets, those thrombocytes are responsible for clotting, along with another protein found in the blood that's called fibrinogen. Okay, fibrinogen is a protein that whenever there is damage, it turns into fibrin, which creates this mesh, and the mesh traps platelets, traps blood cells, it traps everything in there in order to form a clot in the blood. Okay, so this is what happens when, if you cut yourself, vessels are damaged and blood is trying to leak out, clot formation happens. Immunity, we'll go into more of the specifics of this, but I want to make sure you know that white blood cells are also called leukocytes. Again, this is just terminology. Leukocytes are white blood cells. They are made in the bone marrow. Most of our blood cells are made in the bone marrow. Specifically, remember we talked about the hematopoiesis in the bones, specifically the long bones, and then obviously when we're in the red marrow. When we're a newborn, most of our bones have red marrow, but as we age, we get older into adulthood, red marrow is mainly found, in the flat bones of the skull, in the pelvis, in the proximal humerus, the proximal femur. Those are the big blood producers of the body. With white blood cells, we'll get into a little process, something called diapedesis. We'll get into more of this a little bit more later on. This is the process through which white blood cells leave the vessels. Okay, we'll get into more details in a little bit. So looking at what makes up blood. So obviously it's a fluid tissue, the only one. The matrix of this specific connective tissue, we call it plasma. Okay, so basically all of the stuff, when we take out the erythrocytes, we take out the red blood cells, take out the white blood cells and the platelets, and we're just left with the plasma. So none of the big things are in plasma. We take all that out, and that is what we are left with plasma. Okay, plasma is mostly water. We'll see that in a little bit. And this is actually the process by which you can donate plasma as well. There's lots of good stuff here within the plasma that people need, which is why it's often paid for donations. Okay, so the matrix is the plasma. These cells are or at least like the living blood cells, are called the formed elements. On all of these formed elements, the cells are suspended in that plasma as they go through the vessels. When we talk about the formed elements, we have erythrocytes, we have leukocytes, which, little information, this is just a peek ahead of what we're going to look at. And then, of course, our platelets or thrombocytes. Okay? Again, we talk about plasma is the matrix. Formed elements are the other parts, the other big cells floating inside of it. When we talk about blood itself, there's a certain formula, a certain recipe that we want to follow to make sure that our body is good and healthy. Like how much of each of these is normal and how much do we need. So we have something called hematocrit. Hematocrit is the measurement. So you take the blood, you spin it, and you look, at plasma versus the formed elements, specifically the red blood cells, and say, okay, what percentage of the blood do the red blood cells make up? That is our hematocrit. For males, it's about 47% of blood. Females, it's about 42%. Anything above or below is not good. Okay? Below, we call that anemia. It means you don't have enough red blood cells, and we'll take a look a little bit kind of what anemia, what's a condition that can cause anemia. You'll talk a ton about anemia when you get to pathology courses, as well as your lab diagnostics courses. They talk lots and lots and lots about anemia. So those are normal values for our red blood cells. White blood cells and platelets are found in what is called the buffy coat. And then finally that plasma is 55%. So think about 45% of our blood are the red blood cells, 55 is the plasma, and then a little less than 1% is the buffy coat. Here's all of the stuff that make up plasma. I went ahead and took all of that and wrote it out here so it's a little bit more digestible. So please take a look at this. This is something, plug into Quizlet and go off of this. You just want to remember the names when you're looking at this. Say, okay, kind of what can we find in plasma? You'll probably have a test question or two specifically talking about some of the more important parts of plasma, being most of it is water. Albumin is a protein produced by the liver. That's super, super important. Fibrinogen for clotting. And then as well as some of those nutrients and other stuff as well. Those are the big ones to focus on. Now, getting a little bit more technical, this is a question that is very, very often is asked on tests. Teachers love this question because serum, plasma, it gets confusing. So serum is basically plasma minus the clotting factors. Okay? So you think like the fibrinogen specifically, and then any additional bits and pieces of anything that like potential platelets or anything like that, primarily fibrinogen. Let's call them the clotting factors, the things that help with clotting. You take those out and you're left with serum. So serum and plasma are almost identical. The thing you need to remember is that serum does not have the clotting factors. I guarantee you that is going to be a question on a test somewhere. If not in this course right now, depending on what most teachers have a question bank that they pull questions from. So if you don't get it this trimester, you will get it in the future. Okay. That was the plasma, that was the serum. Now we're going to talk about the formed elements, the red blood cells, white blood cells, and platelets. This is kind of our general information. I'm going to dive into each of these. So white blood cells are the only what do we call complete cells. That means that they have all of the organelles, they have a nucleus, they have all the kind of functioning stuff, all the stuff in there that we've learned that cells have up to this point. Okay? So white blood cells within blood, white blood cells are the only complete cells. Okay? That's because so the red blood cells don't have a nucleus or other organelles. Okay? They are not, their job is not to be a full functioning cell, have all this stuff, because they don't last very long. Same with platelets. They don't last very long. And also their job is super simple. Because of both of those things, the body doesn't expel all of this energy to have red blood cells, have a nucleus, have organelles, have all that other stuff. It doesn't fit their job description. Okay? White blood cells do because they're a little bit more complex, but we can see down here, red blood cells live for about 120 days, and then they are broken down by the spleen and then reprocessed, and their materials are reused to make more red blood cells. White blood cells can go from 1 to 12 days for a lot of them, or up to years. Okay? We can have white blood cells live for years and years and years and years. That's why vaccines are effective. When you give somebody a vaccine, like the chickenpox, for example, or if somebody has the chickenpox once by themselves, their body, those white blood cells that fought the chickenpox are then stored within the body, and they're there continuously, and then if that virus ever pops up again, they already have a game plan, and then they can take care of it before we get sick. So some of those white blood cells can live for years. Platelets only last for about 7 to 10 days. And again, what's the other name for platelets right now? Please, please, please remember this. Thrombocytes. That will come back many, many, many times. Now, most blood cells originate in the bone marrow, and they do not divide, okay? Our blood cells are coming from those stem cells, and they're forming from those stem cells, and they're differentiating. Here's just, again, a little picture of some of these formed elements that we're all going to go over. We have platelets, the red blood cells, erythrocytes, lymphocytes, monocytes, basophil, neutrophil, eosinophil, and then a band neutrophil. Okay, we're going to go over all of these one by one. Starting with erythrocytes, the red blood cells. I'm going to point out the things that are bolded are the things, bolded and highlighted are the big things, the things that you 100% need to remember in order for not only for testing in this course, but also for general knowledge. Okay? So erythrocytes are small, and then they contribute to gas transport through hemoglobin. The shape, it's a biconcave disc shape. It is anucleate. That means it does not have a nucleus and essentially has no organelles. It's filled with hemoglobin for gas transport. We'll take a deep dive into hemoglobin in a little bit. And also important to note that red blood cells diameters, all the red blood cells are the same size. Some of them are actually larger than some of the smallest vessels that we have, the capillaries. They contain this protein called spectrin. Spectrin allows the red blood cells to be very flexible in order to fit through the smallest of vessels. We're going to come back to that in a little bit. When we talk again, we're going to dive, the blood's main job, specifically the red blood cell's main job, is oxygen transport, because our body needs oxygen in all the different places to survive. So the shape, the biconcave shape, gives a bunch of surface area in order for gas exchange. Hemoglobin makes up 97% of the cell volume if we don't count water. So pretty much all of the cell is hemoglobin for binding oxygen. They don't have any mitochondria. So all of their energy production, that ATP production, is anaerobic. Anaerobic meaning that they do not use oxygen. So I don't think we've quite reached that point in biochemistry, but that means that they're only doing glycolysis. Okay? And once again, a little note that human red blood cells survive in circulation for about 120 days. That is going to be a test question. They love that test question about how many days does the red blood cells last. 120 days. That comes back several times. Here's just some of the pictures of that biconcave disc shape. This is what a healthy red blood cell should look like. Now, talking about that flexibility here, the spectrin allows these red blood cells to get squished, to bend, to move, to be flexible, and to come back to their typical shape as they're going through all of the different vessels. So here's some examples. If they're smaller, these red blood cells, they can bend super easily to be able to fit all through those. Remember that name of that protein, spectrin, because that's going to be a test question. Now we talk about hemoglobin. Red blood cells have tons and tons and tons of hemoglobin. Remember, 97% of their entire cell, not counting water, is hemoglobin. We'll also talk about hemoglobin a bit in biochemistry, so make sure we pay attention here, because you're going to hear about it, or actually you should have already heard a little bit about hemoglobin in biochemistry when we talked about proteins and how proteins are built. So hemoglobin is a quaternary protein. Take a minute, think, remember what that means. What that means is that there are 4 individual polypeptide chains that all fold, they turn into their tertiary form, and then they come and they all fit together nicely. So it's 4 proteins, basically, 4 polypeptide chains that all come together to make one big protein. And each of these has iron at its base. And so because we have 4 parts of hemoglobin, each of these can bind 1 oxygen, so each individual hemoglobin can bind 4 molecules of oxygen. Here are some normal values. So this is typically, you have 13 to 18 grams of hemoglobin per 100 milliliters in males, 12 to 16 grams per 100 milliliters in females. A lot of times they like to ask the normals, so make sure that you know those. Generally around that 15-ish is kind of the safe number. So the hemoglobin is a red heme pigment bound to the protein globin, so hemoglobin. and it's iron that gives it that red color. We can see in these pictures as well the 4 polypeptides that it makes up hemoglobin. 2 of them are beta, what we call beta chains. The other 2 are alpha chains. Okay, so we have 2 beta parts, 2 alpha parts, and they come together. Don't give that too much thought. Just know, there's 2 beta, 2 alpha. It comes back and loops back a little bit why we mentioned that. There's the beta chain comes together, alpha chain, and they all fit together with iron at their base. So hemoglobin is specifically designed to carry oxygen. Okay, that's its job. But it also does a couple of other jobs. Okay? And it can get into trouble for trying to do its job. So these are terms for the hemoglobin when stuff is bound to it. So we have oxyhemoglobin. So that's hemoglobin with oxygen bound to it, and that is a reversible binding, meaning that they can come apart. It's completely normal, everything's good. Then we have carbaminohemoglobin. That is hemoglobin with carbon dioxide bound to it. Again, not an issue. Carbon dioxide is completely natural in the body, and our hemoglobin helps take carbon dioxide from the cells, brings it up to the lungs for us to get rid of as waste. So completely normal. The issue becomes when we have carboxyhemoglobin. Okay, this is with carbon monoxide. If you've heard of carbon monoxide poisoning and how scary that can be, that's what this is. When we get carbon monoxide that binds to the hemoglobin, it is irreversible, meaning that the carbon monoxide locks onto there and it does not let go. And then we don't have a way for that red blood cell to carry oxygen. We're going to get more into that in just a little bit. We're going to start from the top and work our way down. Okay, so we're going to talk about the oxyhemoglobin a little bit more first. So the oxyhemoglobin is with oxygen bound. And I want you to look at this picture just a little bit right here. Notice how the little blue is supposed to be the oxygen. When oxygen binds to hemoglobin, notice how everything kind of shifts and changes shape. This is what happens to all cells, actually. I should say to all proteins when something binds. It goes through what's called a conformational change or a shape change. Its shape changes slightly because when you add something else to it, like an oxygen, has chemical properties to it. And we remember that our proteins are held together by hydrogen bonds, and they're connected, and they're through a bunch of other forces like van der Waals forces and stuff. And then all of the different components interact with one another for it to make its proper shape. When an oxygen binds, it changes now the chemical properties in that area. And so it creates a shape change where the whole molecule slightly changes shape. And this is exactly what's supposed to happen. When the first oxygen binds, the shape of the molecule changes, and the second oxygen binding site is then open. So that way another oxygen can come and bind. When that second oxygen comes and binds, the overall shape changes again, and that opens up the third binding site. Oxygen comes, binds, it goes through and changes its shape, and then it opens up another site for oxygen to bind, and finally the fourth and the last oxygen is able to bind. And then the same thing happens in the reverse order. One oxygen leaves, the shape changes, the next oxygen is able to leave, the shape changes, the next oxygen can leave, shape changes, the next oxygen can leave as well. So see, that's kind of the process, the way that oxygen is able to bind to hemoglobin. Carbaminohemoglobin, this is with our CO2 binding. Now, CO2 is a completely normal waste product, and it goes and it can bind to the hemoglobin to get carried back to the lungs to be expelled and released. Okay, it is important to note that most of our hemoglobin, I know that I wrote it down here, most of our hemoglobin is not most of our carbon dioxide is not transported by hemoglobin. About 20 to 25% is transported by hemoglobin. The other 70% travels in the plasma as bicarbonate. That's called the bicarbonate buffer system. That's in the future. It's briefly mentioned a little bit when you talk about buffer systems in Biochemistry 1. You'll learn a little bit more about bicarbonate buffer system in physiology next trimester, but don't worry too much about that. Okay, about 20 to 25% of our carbon dioxide is transported by the hemoglobin. Finally, the not so good stuff. This is where we're going to end this first part talking about this. So hemoglobin has a much, much higher affinity or likelihood of binding or desire to bind for carbon monoxide than it does for oxygen. So it wants to bind 200 to 300 times more to carbon monoxide than it does to oxygen. So it binds incredibly strong to carbon monoxide, and you get what's called carbon monoxide poisoning. So this is not this information isn't included specifically in this course, just this part where it's way more higher affinity, but I think it's super important to recognize these signs now and know what this is going on here. Is this is a very, very important thing to be able to notice, especially if you're living in your own apartment or by yourself. So most common sign of carbon monoxide poisoning is you'll see cherry red skin. Now this can be any part of the skin, okay? You can see it in the face, in parts of the body, especially in the gums, in the lips, in the cheeks, fingers, those kind of places. You will see this discoloration, this bright red coloration. Also, you can have headaches, dizziness, nausea, fatigue, confusion, any sort of the signs that you would think that you could get with a lack of oxygen. That's going to be linked to carbon monoxide again, because it's taking the place of oxygen, so your blood can't carry the oxygen it wants to or needs to. We're almost done with pathology, so we're going to do this next one. Now, pathology, another pathology is sickle cell. Okay, so sickle cell is a disease where one part of the DNA, the gene that codes for hemoglobin, specifically the beta hemoglobin, the beta chain part of hemoglobin, is mutated. Okay, there's a single nucleotide substitution. So basically, one little part of DNA got cut out, and the wrong part of DNA got plugged in there. Or the wrong component of DNA got plugged in there. And so now, when it gets built, it's building a non-functional hemoglobin part. It's building a chain that isn't able to do its job. What this causes is that the hemoglobins kind of become sticky, and they sticky to one another, and so the hemoglobins within the red blood cells actually kind of get stuck together, and they form this big clump. This is called polymerization. They polymerize, they stick together, and they form these clumps, or the clumps are known as aggregates. And when you get these aggregates of the red blood cells, it makes the shape of the red blood cells different. Okay, they become misshapen, and because of the wrong shape, they can no longer do their job. Among other issues, the blood can be thicker, so it doesn't move as well as it can get stuck in some of the smaller vasculature because they can't because they have the big clumps in them, they can't bend as nicely. So we can see kind of these sickle cell blood cells, how they look. Instead of a nice biconcave disc, they're kind of flat like this. They're just misshapen. Now I want to break this down. So you have two different types of what you would call sickle cell conditions. You can have sickle cell trait, or you can have sickle cell disease. Because this is linked to our DNA, you can pass this disease on to children. Okay, so when an adult has this kind of condition, they can pass it on to their kids. If you have, because everybody has 2 of each chromosome, if you have 1 copy of hemoglobin that's mutated on your chromosomes, and then a normal copy, you get sickle cell trait. So this is where some of the red blood cells are sickled, and other red blood cells are completely normal. Okay, so they don't typically have full-blown symptoms, but you can have some minor issues with this. You might notice that somebody with sickle cell trait maybe can't run for as long. They don't quite have the same ability to deliver oxygen to the parts of the body that need it. So sickle cell trait also is not it's not all bad. It actually is beneficial against the disease malaria, because the disease isn't actually able to attach to the sickled cells in order to reproduce, and it offers partial immunity to malaria. And that's one of the reasons why sickle cell is very common among people of African descent, because in a lot of areas of Africa, you have high malaria cases, malaria incidents. And so because it was beneficial to have sickle cell trait, more of those people survived and actually passed on the trait, passed on the gene. Now, sickle cell disease, this is when you get 2 copies of the mutated gene, 1 from each parent, and most, if not all, of your red blood cells come out like this. These cells are sick. They don't last very long. Instead of lasting 120 days like the healthy cells, they only last about 10 to 20 days. That leads to anemia, which is not enough red blood cells. It leads to an inability to pass oxygen, and this can be a serious condition for these kind of patients. So you need to make sure, if somebody's presenting with these kind of symptoms, and then also combined with their medical history, their parents' medical history, and you want to take into account all parts of the patient when you're looking at potential diagnoses. This is always something to keep in the back of your mind. It's not super common that we as chiropractors are going to see something like this, but it may happen every now and then. Okay, we're going to go ahead and end part 1 right here, and we will continue in part 2 with the white blood cells, or the leukocytes.